Hello, and welcome to Inside Cancer Careers, a podcast from the National Cancer Institute, where we explore all the different ways people fight cancer and hear their stories. I'm your host, Oliver Bogler from NCI's Center for Cancer Training. Today, we're talking to three computational scientists working on cancer in NCI's Center for Cancer Research. They've recently published an AI tool to help clinicians predict response to immunotherapy, a great example of how people with all kinds of skills can make important contributions to ending cancer as we know it. If you recall, in our first episode of Season 2, we talked to Tony Kerlavage and Jeff Schilling, informatics leaders at NCI, about the general impact of AI on cancer research. Today, we're highlighting one of the teams at NCI working with AI and exploring their patient-focused work. Listen through to the end of the show to hear our guests make some interesting recommendations and where we invite you to take your turn. So with us today is Dr. Eytan Ruppin. He's the chief of the Cancer Data Science Lab at NCI. Welcome, Eytan.
Thank you. Thank you so much.
Also with us are two fellows in Dr. Ruppin's group, Dr. Tiangen Chang. Welcome.
Thank you, Oliver. Glad to be here.
And Dr. Yingying Cao. Welcome to you.
Thank you, Oliver. With pleasure.
All right, Eytan, what is a cancer data scientist and what role do they play in cancer research in 2024?
So, as you may know, Oliver, now in cancer research and also in treatment, we are generating a lot of data spanning different dimensions in a way that is exponentially increasing every decade compared to the past. And that, like in many aspects of our life, creates a large amount of data that really needs computers and computational analysis to try to make sense of it, both to understand what's happening in cancer from a basic science research point of view, and to try and devise ways, which is the specific focus on my lab, on trying to sift through the data and try to decipher what are the essentials that can help us advance cancer therapy, both in terms of identifying new targets, new combinations to treat patients, and in terms of predicting the patient's response to an array of possible therapies, aiding the physician, of course not replacing the physician, but aiding the physician to make clinical decisions and to choose the best matching treatment to any given patient.
So a great example of your work was recently published. Tiangen and Yingying, you are the lead authors on that paper. It's focused on immunotherapy, a very fast-moving and important and promising area in cancer research right now. Please tell us about that work.
Sure. So we all know that cancer immunotherapy has been a game changer in the cancer treatment over the past 2 decades. But here is the thing, not every patient responds to this therapy. So on average, only 20 to 30% of the patient will see a positive response. So there is an urgent clinical need to really figure out which patients will eventually benefit from this therapy. So Loris is a machine learning model to help predict patient response to immunotherapy. And it is a logistical regression based model. It takes 6 variables of a patient as input, and these variables are routinely measured in current clinical settings. So the output score of Loris can reflect the response probability of a patient to immune checkpoint blockade therapies. So we have validated this model in a lot of currently existing cohort, but we still want to see it to be further validated by more clinical cohorts in the future, and finally benefit more patients from this therapy and help clinicians to make better decisions.
So Yingying, how did you come to build the model in the way that it is built? How did you choose the variables that Tiangen mentioned? How did you put them together in a way that helps the clinician?
The way how we choose the features is based on the how many features are collected for each cohort. The most common features are the 8 features, and we choose 6 of them to build our model. So because it's a model for clinical use, we want to build a model that can be easily to use for the clinicians. So it's more easier collected the features like the booming collected from the blood and the tumor mutational burden. So they are routinely collected during the clinical usage.
So these are readily available data items
Yes,
that most clinicians will have.
yes.
Explain to us what is logistical regression or logistic regression. Did I say it wrong? I'm not a
Yeah,
data scientist. Have you noticed?
yeah, it's correct. Yeah, absolutely correct. So yeah, logistical regression is a kind of regression. So it's we know that linear regression, right? Y equals ax plus b. This is a linear regression. However, we know that the response rate is not from infinity, right? It only have a range from 0 to 1. So you need to convert the from the negative infinity to positive infinity into a range of 0 and 1. So here you need to make the exponential and the division. So here then you make the logistical regression. It is essentially a linear regression, but just convert the value range from infinity to 0 to 1. This
Okay, I'm going to pretend that I understood that and ask a follow-up question and say, so how can I imagine it? I imagined, and I've seen the tool. It's online, right? Anybody can go to the web page, and we'll put a link in the show notes. You can put in these data elements, and then is the model weighting them differently and combining them into a score?
exactly. So yeah, you can input this. I mentioned the 6 features such as patient age, the tumor mutational burden, the previous therapy history, and so on. So you have the value for these 6 variables. Then you just input these variables, and there is an equation. But although you cannot see it, but it's in the background. Then you click calculate. This equation will take these 6 variables as input and give you a value between 0 and 1. Say the output is 0.5. That means this patient have 50% opportunity to respond to immune checkpoint blockade therapy.
Okay. Now you mentioned their mutational burden. I imagine that as the sort of a measure of the number of mutations or the frequency of mutations that you have observed in the cancer. That comes from sequencing data.
Exactly. Yeah.
Okay. And is that one of the more powerful variables in your model, or is it right there alongside some of these other features?
Oh yes, there's a little bit background. So this tumor mutational burden is, I think, the first FDA-approved biomarkers in cancer. So to quantify which patient can receive these immune checkpoint blockade therapies. So FDA gives a threshold of 10, so that for patients, if they have a tumor mutational burden of 10 mutations per megabase on their genome, they will be qualified to use this immune checkpoint blockade therapies. However, later researchers found that this is a suboptimal biomarker because it will miss a lot of real responders. That is, there are a lot of, so in our study, we found that maybe there are more than 60% of the responders, they have a tumor mutational burden less than 10. So this biomarker is quite suboptimal. And this is why we developed LORIS, and from Loris, we found that we can identify patients that are previously thought to be poor candidate for immune checkpoint blockade therapies, but they are actually the responders. So we hope this tool can help clinicians to select more patients that can benefit from this therapy.
The model that you developed is based on historical data, right? On retrospective data that you had. And I think you mentioned in the paper that you need now as the next step or one of the next steps a prospective study to really test this. So tell us about that. What would that look like?
Yeah, so we have basically in clinics, we have 2 types of data. One is retrospective data, one is prospective data. Retrospective data that is collected before, that is already there. So you can actually you can do cheat, right? Because you already see the result, so you can modify your classifier so to fit the result. So you can cheat. However, for the prospective data, you cannot do that because the data is still not there. So you set up a new clinical trial, and then you have the model first. Then you collect data to see if your model really work or not. So prospective data is really important in computational science. So that can evaluate if a biomarker or a finding is really true or not, because in this case, nobody can do the cheat.
Interesting. So Yingying, a different question. I don't know. When I looked at the paper, I read the paper and looked at the model. It struck me as surprising that only 6 pieces of data can be so powerful in that prediction, and that may be because I lack mathematical intuition. But were you surprised by that? We always talk about cancer as so complicated. There's so many things happening.
Yes, I think it's quite surprising because intuitively in the machine learning study, the more features should have better performance. But currently, we found that these six features with the current data have the best performance. I think it's not intuitive. It's surprising. The reason is that firstly, usually the clinical data is quite noisy, and when the data is quite noisy, not it doesn't mean that the more complex model or more features we have for the uh, better results. And another thing is that it doesn't necessarily mean because of the noise of the data and the more features uh, can can cause overfitting. This is also a major, I think the major benefit of our model that it can be generalized to the test data and the data set from other cohorts. So I think it can be say surprising and it's not surprising because we can explain that.
I would like to pitch in just briefly. So first of all, Oliver, the simpler the model it it's the better and it's more believable. Okay,
Okay,
so simple is an advantage in the parlance of models, complexity, machine learning, what have you. It's called Occam's razor.
sure.
So simple is good. That's one thing. Second of all, a lot of features are strongly correlated between them. So the fact that you see only a few gives you a lot of information, hidden information also on other features. Okay? The question is to pick the ones that are most informative on the state of the system. And that is what very nicely Tiangen and Yingying have done. But thirdly, we should remember that we think that if further tested and validated going forward, this could be valuable. That's our hope, of course, our belief, okay? But we are far from perfect. So the performance, without going into technical details of measurements of performance, is 75%, 80% accuracy, but not more. Okay, and we should remember that,
Okay.
and that's a very important open challenge, and we are not yet there. We are not that there yet.
So it's a really good first step, but there's more to come.
Exactly, and the way I would relate to it, and even if prospectively verified and so on, is another way to help clinicians take the best decisions.
Yes.
It's not that, the futuristic, whatever, the AI medical robots that are now, we are very far from that.
Right. Right. So, Tiangen, the other thing I noticed is that this model has predictive powers in a number of different cancers, right? It's not just for one type of cancer. And I guess that points to a commonality of mechanism amongst these cancers in relationship to the efficacy of the checkpoint inhibition. What are your thoughts on that?
Yeah. Thank you for the great question. Yes. So yeah, we because we collect a lot of cohorts. These different cohorts, they have different cancer types. So our first question is if we can build one model that can capture the commonality between cancer types to get a model that predict the ICB response in different cancer types. So then that is our first model. And then, as you may know, that this model captures the community, but just do not capture the specific special property of individual cancer types. So then we developed the second Loris model, that is specifically lung cancer. We do in lung cancer because this is a cancer type that we have the largest sample. So we want to still keep the statistic power. Then we found that use the same strategy, we can also predict lung cancer immunotherapy response quite well. But the model is slightly different. And in the lung cancer, we add one feature, that is the PD-L1 expression. And this is a commonly used feature in lung cancer. Clinicians already know that the PD-L1 expression is a very important feature that determines the immunotherapy response.
Right. PD-L1 is one of the checkpoints, right? The checkpoint inhibitors try to knock down, so to speak, to allow the immune system to fire. Is that right?
Exactly. Yeah, it is expressed on the cancer cells and stop T cells to kill them. Yes.
Eytan, thinking about the Loris work and the many other things your group does, and you just told us we're not about to have medical robots coming into our hospitals. What are the next years hold for your AI work and AI in general in this domain? What's coming up?
Yeah, that's a fantastic question, and I do not know the answer. I can tell you what we are working on,
Okay.
and excites me, and I'm hopeful about. So we, as my lab, and our collaborators, of course. So we are working on two, and I am gung ho about developing methods to better match treatments to patients. Okay, and I'm trying to do that in two main new dimensions. One of them is, believe it or not, look at the good old 100 years pathology slides, you know,
Mm-hmm.
which are the backbone of tumor classification and via that some treatment recommendations, and actually apply the recent, developments, buzzwords in AI and machine learning and deep learning and all that, and predict the molecular characteristics of tumors directly from the pathology slides. The gene expression, the proteomics, genomic alterations, mutations, what have you. This is now an extremely hot field with a few fantastic labs working in the US, Europe, whatever, and we are also heavily excited and invested in that. Actually, we just published a paper in Nature Cancer today on this topic,
Okay.
and a month ago in Nature Medicine. So you can see the pace of things. And in this paper that we published today, we show that we can actually take the slides and predict omics data from them, and based on that and previous work that we have done, bring treatment recommendations. We are showing validations in 5 different independent cancer types and treatments.
Wow.
So that's one direction. Slip sliding again, Oliver, if you remember Paul Simons. I mean,
Yes.
the kids have no idea what we are talking about, but right,
It's a classic.
the young researchers. So that's one direction, which I'm gung-ho. What can we do from the slides? And many others, of course. There's others doing fantastic work on it, okay? The other topic, which Tiangen and Yingying are actually taking active part in, is asking what can we learn from the blood,
Yeah.
okay, on the tumor immune microenvironment. And so together with other wonderful collaborators in the NCI, we are collecting matched data where we have from the same patients, both the blood and tumor molecular information. We are doing what is called single cell sequencing and what have you and whatever. And we are learning what we can predict from the blood about what's happening in the tumor microenvironment. And for that, for example, predict the response to immune checkpoint therapy. So this guy, Tiangen here, has now a paper finding with some very exciting findings on that, which we are gonna submit in the next few days. And, if I'll tell you, I'll have to kill you, dear. So,
I won't ask.
so anyway, so that's the two main directions where I'm excited about, and I believe that we can deliver meaningful advancements in the next few years. And
So
here, this is recorded, so you can call me accountable, right?
yeah, we'll check in a few years and see how you did.
The virtual site visit.
Exactly. Those are exciting areas. It sounds fantastic. Tiangen, I know I can't ask you specifics about the paper you're about to submit. It sounds really interesting, but is it building on Loris? Is it a natural? No, it's something completely different. Tell us.
Okay, yeah. So yeah, currently actually, yeah, I continue working on predicting and understanding the response to immunotherapy, but now I'm more focusing on studying the tumor microenvironment. So I do not limit myself to predict the immune checkpoint blockade response only from clinical data. So for the paper Eytan just mentioned, so yeah, we use more advanced techniques such as single cell RNA-seq, so we can look into individual immune cells and tumor cells in the tumor microenvironment and also in the blood. So we use a similar strategy. As Eytan said, we computational scientists, so we collect data from public data bases. So we collect a huge cohorts of data. To our knowledge, it is the largest in the head and neck cancer. This is a single cancer type. And we found the B cell signature that can predict the immune checkpoint blockade response quite well. And it not only works in the tumor microenvironment, but also works in the blood. So simply from get the blood of a patient, then do the single cell, or even simpler, do the flow cytometry, we can predict the response probability of this patient to immune checkpoint blockade therapies. Yeah.
Right. So this is a kind of a new phase where you're not using clinical data, you're using more biological data that are perhaps also harder to acquire. They're not routinely perhaps collected in the clinic. But is there a future where the two might converge and allow you to have a more comprehensive prediction model? I mean, Eytan mentioned that the Loris model has an accuracy of approximately 80%. So will you get to higher numbers with this approach?
Yes, we hope so. Yes, one of the advantages of machine learning and this artificial intelligence is to integrate data from different modalities, right? From the clinical data, from the molecular data, and to integrate all this data. So we hope we can increase the predictive power, so to lead us to a more accurate prediction.
Yeah. So I want to mention in that aspect, it's very hard, Oliver. There is little molecular analysis of patients' data yet uh, because of the costs and because it's not part of routine practice. So where we hope that it could be a game changer is these technologies where we look at the slides and infer the molecular data. Okay, and we can do it pretty well now. That can potentially enable us to contribute to democratizing medicine. So, even within our nation, there's huge differences between different regions and so on and so on. It is our hope that if further developed, this could be a very important factor, and I truly believe we can do it. I am actually, together with Ken Aldape, our chief of pathology and my good friend and collaborator, we connected a few foundations that do work in Africa. And we are actually engaged almost every week. We spend time meeting with at least one or two groups enlisting them to get data from Africa, deploy our methods. There's a whole host of problems and challenges. It's a topic for another discussion and interview, but we are persistent people. So actually, I grew up as a kid in East Africa,
Ah,
and I actually don't test my Swahili, but I actually have a deep connection and a moral uh, commitment is a strong word, but
yeah.
whatever interest.
Hmm.
So I'm very excited to see how it will unfold. And our the people we speak here, the medical oncologists and the pathologists in Africa, are really excited about that. So I hope we can move forward and make a difference. It's early days.
But that's
Yeah,
exciting. So the humble H&E slide may put the sequencing companies out of business.
never. That's my plan. That's my plan. The revenge of a lowly federal clerk.
But exciting, as you say, it will make that kind of information available in many more parts of the world where the kind of deep sequencing is still out of reach. That's exciting. And then I understand that those molecular data inferred from the H&E slide could then be combined into predictive models that you're building. Yingying, how does your next piece of work, your next project, fit into this scheme now that Loris has been published?
As you know that Loris uses the features from the clinical routine collected data, and they cannot explain more details in the mechanism of how the immunotherapy response in each patient is, and how the in the human body the tumor microenvironment react to the tumor cells. So I'm more interested in the details of the interaction of tumor microenvironment and the tumor cells. So currently, I use the single cell RNA-seq data and the spatial transcriptomic data to really understand um, the tumor microenvironment part.
Fantastic. I'm so
Yeah.
excited to hear about all the things you're all doing, and I can't wait to see what amazing advances you make. We're going to take a quick break, and when we come back, we will be talking about career paths with our guests.
Cancer researchers, are you drowning in a sea of scientific papers? Wish there was a way to cut through the noise and find the research that truly matters to you? Look no further than Nancy, the revolutionary new AI-powered app from the National Cancer Institute. Nancy isn't just another research publication app. It's your personal research assistant, designed to understand your unique interests and connect you with the most relevant papers, people, and events in the field. Imagine being able to chat directly with research papers, ask questions, and get instant answers. That's the power of Nancy's AI-driven chat with a paper feature. But Nancy is more than just a conversation starter. It's a networking tool helping you discover new connections and build your professional network. It's a learning tool recommending papers you might not have found on your own and helping you quickly get up to speed in new research areas. Simply declare an interest by bookmarking papers on a topic into a folder, and Nancy helps you stay up to date. Whether you're a seasoned researcher or early in your career, Nancy can help you take your work to the next level. By harnessing the power of AI, Nancy is transforming the way cancer research is done. Download Nancy for iOS today from the Apple App Store. That is NANCI, and experience the future of research for yourself.
All right, we're back. Eytan, you did an MD and a PhD in computer science, an unusual combination. How did that happen?
Serendipitously, or whatever you say it in English, right?
Uh-huh.
So actually, my favorite talk to young fellows in the NCI is the title is How Not to Build a Scientific Career.
Okay.
All the mistakes I made, and there are so many, Oliver.
Okay.
You'll be so I started late. I studied medicine. And then at the age of 30, I finished my studies, finished my internship, and I came to the conclusion that I do not want to be a practicing physician. And then what will I do, right? And
Yeah.
so, so I was really thinking, I was quite lost,
Okay.
to be honest. And then I came upon a book because you asked us to think about a book or movie or something that influenced our lives. And that is a book that changed my life.
Hmm.
So the book is called Bach, Escher, Gödel
Oh yes.
by Douglas Hofstadter.
Oh yes, I've read it. Yeah.
Yeah, okay. Did it change your life?
It didn't change my life.
That's an amazing book about the deep connections between computer science to music and art and the concept of recursion, but brilliantly written. And I decided that I want to become a computer scientist.
Amazing.
So at my time, Oliver, dinosaurs roamed the earth.
Yes.
And you remember them, they were big and green and frightening, right?
Uh-huh.
There wasn't an MD PhD or something in my country, so I had to do first, second, and third degree in computer science
Wow.
in parallel to my residency in psychiatry.
Wow.
So I didn't sleep for about 5 or 6 years.
Wow.
And luckily, I worked hard and did it in record time. And then I went to a postdoc and a residency in the US, and the rest is history.
Interesting. Yeah, you founded the Cancer Data Science Lab at NCI, right?
Correct.
And we'll get back to that in a moment. But first, I want to ask Yingying, what got you into science in the first place? Yingying, what was that moment that made you say, I want to do computer science?
During my bachelor study, I studied preventive medicine, and
Okay.
as an, I even don't know what bioinformatics is after I graduate from bachelor
Okay.
degree. I was enrolled into a bioinformatics group during my master degree study, and I found it fascinating that because during preventive medicine study, we focus on the population medicine. We focus on how to make some strategies to prevent prevent the disease. For example, the change the is a living style. So to make some strategies to do on the population level. So we don't do it on the individual level. And during my bioinformatics study during my master degree, I found that each human body can be quantitatively can be a quantitative object. So the each molecules and in the human body and even the abundance of different cell types can represent your immune level. So I found it quite interesting to treat each human body as a quantitative object and to see how the human body can fight off the infectious disease. In master degree, I studied the virus infectious disease. Actually, the cancer research and infectious disease, they share very common things, especially during in the immunity. So I found that the immunity is a very fascinating subject, and and then I enter NCI and to study the cancer research, the cancer immunotherapy, and I found it. Yeah, I think every step is unexpected, and when I studied, I found it interesting, and so I don't know what the next step is, but I found I'm more and more interested in the scientific research, and I want to build my career on the scientific research to be in the future maybe a mentor, because during my scientific path I meet a lot of very good mentors. I want to transfer what they teach me to the trainees, and also I also have a lot of ideas even now that, but I cannot do it alone. So I want to build a scientific team to include different talents and to collaborate and find the truth about the immunity, the cancer research, and even some infectious disease.
Thank you for sharing that. So you kind of immunity first, and then you pick up the tools you need
Yeah.
as you go along. Tiangen, how about you? What got you interested in science and computer science?
Sure. I think, yeah, this is a great question. And the answer for me is simple. I think it's largely due to personality. So when I was young, I can spend a lot of time sit down to work on a problem for a very long time. And I was not very good at playing games or socialization with people. And I enjoyed reading, walking in the nature, or exploring science. So I don't like doing repetitive work, but I found great joy in spending a lot of time thinking about a problem and eventually finding the answer. So that moment of discovery makes me so happy, and I enjoy that feeling immensely. So actually, I started my PhD career in plant science. Before that, I have a bachelor degree, and at that time, I knew little about biology. So I didn't find much different between animal science and plant science. My PhD supervisor was wonderful, and he taught me two very important lessons that I still benefit from it. So, firstly, you need to trust yourself and focus on important questions. Do not just follow others. And secondly, focus on translational oriented research. I'm not saying that basic research is not good. Actually, basic research is extremely important. But you know, every supervisor has their own style, and this style will influence greatly on their trainees. And at 2021, I decided to study cancer. In this year, one of my uncles diagnosed with cancer, and before that, two of my grandfathers also had died of this disease. So I decided to devote myself in fighting cancer. And I always feel very fortunate to have the opportunity to get into this field. So I feel very thankful to Eytan, and even now I don't know why he chose me, because at that time I was a completely clueless guy. Maybe that day he was drunk, but that it was a really beautiful mistake. And I also want to thank Yin, and she joined the Eytan's lab a little bit earlier than me, and he introduced Eytan to me. So yeah, this is my story.
Thank you for sharing that. I mean, this is really interesting to me. So all three of you really started in biology and in medicine in different parts of that ecosystem, but came to data science more out of an interest or a recognition that that tool set and that way of approaching a problem could be interesting and helpful, right? Is that, did I get that right?
Yes, but not exactly in my case.
Okay. I mean, you got lost and then you found yourself.
I'm still drunk, according to Tiangen. So no, so actually, my career has been so tumultuous. So I actually, because I did my residency in psychiatry, I was naturally interested in understanding what's happening in the brain, in neurological and psychiatric disorders. So I actually spent my first 15 years of my career as a computational neuroscientist, building models, neural network models, trying to understand what happens in Alzheimer's and schizophrenia in the brain. And after, I don't know, I was already a professor of medicine and computer science, accomplished, blah. I came to realize that I will not understand it. I don't believe that in my lifetime anyone else will understand it. So I decided to take the bold step and reinvent myself completely and move to a much easier problem: how to solve cancer.
Fair enough.
That's true.
Yeah.
That's true.
Well, and how's it going?
Not so bad. I mean, surprisingly good. I mean, you know, our ability to understand what's going in cancer and to come up with better treatments is advancing, I think, objectively or not objectively, at a much more impressive pace than what we understand about the brain and how the brain works.
Yeah. Yeah.
So I'm very, very happy that I finally made one good choice in my career. Yeah,
I'm sure you made many, but we're very excited that you chose cancer because we need incredible people like you and your team. So I wonder, as my final question, what is your advice to people who are listening who might be primarily interested in biology, medicine, cancer, and be thinking about, well, how do I continue to do that in the current AI revolution? Or conversely, coming from the computer science side and thinking about how do I come into the world of cancer? I want to do something really important and meaningful in my life. What's your advice to people, young people thinking about these questions?
that's a wonderful question. I'll be happy to give you my take, but I want first to give a chance to my fellows
Sure,
to answer it, and then I can take it from there.
please.
Yeah, I think, yeah, from my experience, you need first to choose a important question, so research question. Then you just focus on this question. I still remember that 3 years ago when I planned to join Eytan's lab, I prepared a research plan. So I just want to appeal to attract Eytan, so to see how beautiful this plan is, how thoughtful I was, and so he can choose me. But yeah, now when I look back, it's very naive. Just as Ethan said, all is very good. The only problem is that it cannot be real nice. So, I found that still the question is still there. So at that time, I just want to work on the cancer immunotherapy and the tumor microenvironment, and I am still working on it, and I will work on it for the next 5-10 years. So I think the scientific question is really important, but the method will change a lot because with the advancement of measurement and the computational tools. So uh, for me, I never put a major of my attention on the technical things. I put more attention on the scientific questions.
Yingying, what's your perspective?
Yeah, I agree with Tiangen that I think the to I think the scientific question in your mind that puzzle you as the motivator you is very important. If you don't have that question in your mind that you want to solve it, it's difficult to continue because you will meet a lot of challenges and difficulties during the scientific research. If there is no scientific question that puzzles you and can make you sleepless, you can it's difficult to continue. So I think it's very important to find a real interesting question. For me, I found it really fascinating, the tumor microenvironment, and I'm very lucky. I think this area is uh, is a very lucky area for bioinformaticians because more and more advanced techniques and currently the spatial techniques can give you the spatial information of the molecules and the cells locations and even the distance between different cells and is unbelievable maybe 20 years ago and even people are interested in it. It's difficult to study that in such details and also we can do it in single cell level and the spatial level and so I think more and more advanced techniques are can help us to understand our questions in our mind. And there is one part I don't agree with Tiangen is that Tiangen don't think the techniques develop algorithms or techniques are important. I think the algorithms and the techniques are very important, and the techniques are very important. But the algorithms are even more important because we need to understand more reliable to interpret the data. If the tool or the algorithms we rely on is not um, reliable, and the interpretation of the data, interpretation of the biological system is wrong. So I think that if I develop a team in the future, I will focus on the algorithm development and also the biological part.
Thank you so much. Thank you. Eytan, what do you say to people thinking about a career in your area?
Yeah, so I will answer at a higher level. Okay. Uh, at my rank, we are clueless, so we only talk at a higher level, right?
Of course.
So, seriously, only choose this career if you love it. If you're excited by science, if you're excited about the cause, listen to your heart. Right? Like in any decision we make in life,
Hmm.
you can think from here to the moon and back, but listen to your heart. Second of all, be a nice person. It is so important for your success.
Hmm.
Science today is a team work, both in running your own lab and creating a good atmosphere. I always say I only hire and work with nice people, okay, because I'm not paid enough as a lowly federal clerk to work with not nice people, you know? And also with the collaborators. It's so much emotional IQ, how it's called, in leading, not less important than every other aspect. So be a nice person, an honest person, a good collaborator. We are at the data science lab when I was hired by Tommy Sterling. He asked me, what's your vision, Eytan? And I said, we will be a center. We will help people. And I believe we do. Okay, we are not only doing our own research, of course we are, but we are helping a lot of other people, and other people are helping us. So that's the second thing. First one was love. The second one was goodness and kindness. And the third one is bravery.
Hmm.
Be brave. Okay. Pursue important questions. Don't strive, especially it's a tendency, I don't know, in young PIs or whatever, to publish a lot. That's stupid. Try to do something meaningful. And that is especially true in CCR at the NCI. Okay. Our mandate, explicit mandate, as you know, Oliver, is to pursue high risk as much as we can projects and be persistent and relentless and try to solve them, on a long range. So just to close circle, I told you that today we published a paper and so on. This paper actually, without going into the details unless you ask me, closes 12 years of work, of a continuous work in my lab on a specific challenge,
Wow.
which started in 2012. Our first paper was published in Cell, so not in a trivial journal, in 2014. Since then, we have been working tirelessly to try and improve that. Okay, so the starting point was published in Cell, okay, but wasn't good enough. And so, luckily, I always say to my fellows, we are so bad that we can always improve. Okay, so because the challenges are so big.
Yes.
Okay,
Yeah.
so 12 years of research. I was just telling my wife this morning. I will not mention her name because she will kill me. Uh, that we published this paper and it's 12 years of the fruits of 12 years of work. So that makes me feel, take a pause and think about it.
Yeah. 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 Fantastic. Well, thank you all for that advice. Really, really good and thoughtful. So it's time for our segment now that we call Your Turn, right? Because it's a chance for our listeners to send in a recommendation that they would like to share. If you're listening, then you're invited to take your turn. Send us a tip for a book, a video, or a podcast, or something that you found interesting or inspirational or amusing. You can send those to us at ncicc@nih.gov. Record a voice memo, send it along, and we may just play it in an upcoming episode. But I'd like to invite our guests to take their turn. Let's start with you, Yingying.
I will introduce a very interesting podcast. Its name is Intelligence Squared US. I don't know whether you are familiar with it. I found that it's very interesting. It's about the debate on the important topics, including some science and technology and the society and the culture, and I think the different topics as you can imagine in your life. And I'm very interested in listening in listening to the debates. I found for one topic um when they raise the topic, I have some opinions in my mind. But after I listening to their debates from different part, I change my mind sometimes, and the topic become more clear to me. I think this is very familiar with the scientific research. Yeah, yeah, this is what scientific research make me interested because I always want to solve puzzles and make everything in my life more clear. So for example, if I meet something or some challenges in my life, I want to make it. I want to understand it and make it clear. And the intelligence squared US is a debate that can make things clear, and it make you think twice and always step back and think twice, step back and think twice, and make this clear. And I think it's a very interesting podcast, and people can learn a lot. And
Thank you. Thank you for that tip. We'll put a link in the show notes. Tiangen, what have you got for our listeners?
I would like to recommend a book, and the title of the book is Worldviews: An Introduction to the History and the Philosophy of Science. The author of the book is Richard DeWitt, and this book is a great read that takes you on a journey through the evolution of our understandings of the world from ancient times to the modern day. This book really blew my mind by showing that how different worldviews can lead to completely different understanding of the same universe. And after reading this book, I have had a feeling that even now, most of our scientific findings could still be wrong, but they are meaningful because they represent steps towards a better understanding of our world in the future. Just like a famous saying that all models are wrong, but some are useful. So yeah, finally, I want to highlight that what is really cool about this book. So this book just breaks down very complex ideas in a way that is extremely easy to follow. So it is perfect for anyone who is curious about our world, science, and its history.
Thank you. That sounds really great. I might just pick up a copy. That sounds really interesting. Eitan, your turn.
Yeah, so I just work, guys. I don't read anything. I don't listen. I have no time. I don't understand. I'm a sucker of popular science and science fiction. So, a very large genre. And I don't know if you know Isaac Asimov, who is, and so on. Actually, in addition to being a fantastic science fiction writer, and the series of the robots
Yes.
other series, he also wrote fantastic popular science books,
Hmm.
really fantastic. And I would, once when I had time, I would read them. What I do now is I subscribe to New Scientist.
Mm-hmm.
New Scientist is in my mind a fantastic popular science. I read a lot. I'm a aficionado of physics, and in another world, I would like to be a physicist. Somehow it didn't wasn't in the cards for me in this life, but maybe in another life, because I'm so fascinated. And our youngest son studies physics, so I
Oh,
live vicariously by him. And my favorite thing is to watch different physics, popular physics shows in series with him whenever he comes home. And then he explains to his stupid dad, very complex concepts, and he's very patient with me. And I'm so happy and I feel so proud. I'm so happy to be stupid with him. You see what I mean? And I live vicariously.
Yeah.
So I recommend New Scientist. I'm not a shareholder. I
No
have
worries.
no conflict of interest. But I love popular science.
I think that's a really good recommendation. Thank you very much. Well, thank you all three of you for spending time with us and coming on the pod and sharing your insights and about your exciting work. I wish you all nothing but the most tremendous success. You're really doing such important work, and so thank you.
I want to thank you really very much for having us. And I'm not just being polite. I come from the Middle East. I'm not polite by definition, right?
Okay.
But I always talk sincerely. So I think it was very enjoyable. I hope my colleagues, Yingying and Tiangen, share the same thing.
That's all we have time for on today's episode of Inside Cancer Careers. Thank you for joining us, and thanks to our guests. We want to hear from you. Your stories, your ideas, and your feedback are welcome. And you're invited to take your turn and make a recommendation to share with our listeners. You can reach us at ncicc@nih.gov. Inside Cancer Careers is a collaboration between NCI's Office of Communications and Public Liaison and the Center for Cancer Training. It is produced by Angela Jones and Astrid Masfar. Join us every first and third Thursday of the month, wherever you listen. Subscribe so you won't miss an episode. If you have questions about cancer or comments about this podcast, you can email us at nci.info@nih.gov or call us at 800-422-6237. And please be sure to mention Inside Cancer Careers in your query. We are a production of the U.S. Department of Health and Human Services, National Institutes of Health, National Cancer Institute. Thanks for listening.